Today in this video we are going to discuss about the subject biotechnology, in which we are going to see particularly about the recombinant DNA technology and how genetic engineering happens through recombinant DNA technology. So this recombinant DNA technology was accomplished by Stanley Cohen and Herbert Boyen during 1972. So by their strenuous effort, they found out the recombinant DNA technology. By the recombinant DNA technology, we could able to modify the agricultural products and also the hormonal products. For that purpose, this technology is used. So first of all, we'll see what is meant by recombinant DNA. Recombinant DNA, when the DNA of two species are made to combine together, it is called recombined DNA. And when this recombined DNA is inserted into a host cell, then it clones into multiple times. That is the principle involved in that. So first of all, we will see in the recombinant DNA, we will take up the that is the vector DNA and the original DNA or the foreign DNA. The foreign DNA are which we are interested to multiply. That is, in this case, we are interested to take the first of all, we'll see the vector DNA and the foreign DNA. Foreign DNA is the human insulin. We know pretty well the DNA is the genetic material. So this foreign DNA and then here the vector DNA. The vector DNA, the vector. Vector DNA is nothing but the carrier. Vector means the carrier gene. Here the foreign gene, with the help of the endonuclease, or it is always called molecular scissor. That means at a specific place it will make a piece of DNA that is called molecular scissor or endonuclease. With the help of endonuclease, we can take a piece of DNA. A piece of DNA with the help of molecular scissor called endonuclease. Endonuclease. AS means enzyme. Usually ligase, nuclease, DNA polymerase. AS means enzymes. So with the help of endonuclease, we cut a small piece of insulin DNA, foreign DNA taken from the human being. And here the vector DNA is plasmid DNA. By the use of endonuclease, we make a small cut in the circular DNA. This is called plasmid DNA or circular extra chromosomal DNA. Plasmid DNA is the DNA artificially prepared from the bacterium. So this plasmid DNA, a small portion is made to cut, that is called with the help of endonuclease, called or otherwise called molecular scissor. Like that, here also with the help of endonuclease, we have taken a piece of DNA. This is called extrachromosomal DNA. Circular DNA of plasmid, DNA of plasmid, or extrachromosomal DNA which can replicate by itself. That is the speciality of this plasmid DNA. Then what we do with the help of ligase, an enzyme called ligase. So we put this with the help of ligase enzyme, we insert this insulin DNA in the place where there is a space, where there is a slit. So this extracellular DNA, plasmid DNA. So in this place we are inserting this, here we are inserting this foreign DNA or foreign DNA. Then what we do, this is inserted into a host. This plasmid DNA with foreign DNA this is always called recombinant DNA, because we added the DNA of two species, recombinant DNA of plasmid DNA and foreign DNA. This is inserted into the host cell, that is bacterium or Escherichia coli, E. coli, Escherichia coli. So this is chromosome of bacteria, and then here we are inserting this. So what is available in the Escherichia coli? DNA polymerase is available. DNA polymerase, an enzyme. So these are all enzyme 1, and ligase is an enzyme, ligase, and endonuclease. So 3 enzymes are used in this recombinant DNA. And genetic engineering. Genetic engineering means genetically modified the thing we are going to obtain it. So with the help of DNA polymerase, so when we introduce the foreign DNA into the host cell, with the help of DNA polymerase available in the Escherichia coli or bacterium, it multiplies or it clones. So a single cell is divided into 2, that is called cloning or multiplication. Cloning. So like that, again this is divided into 2, like that, and it multiplies many times. So this consists of the insulin DNA is available in this, and this insulin we are making use of it. So this is how genetically engineered insulin is prepared, and recombinant DNA by the method of recombinant DNA. So the recombinant DNA is made by palindrome, that is the sticky, when two DNA is there, it is the sticky end will be there, that we will see separately how recombinant DNA um, is conducted. That we will see in this separate video. So in this video, how insulin is prepared by the usage of recombinant DNA and genetically engineered insulin. So again, to repeat, we will take the foreign DNA and we are making it into a small piece which will contain nucleotide, and for that we use the endonuclease. The same enzyme is used in the plasmid DNA also, to make a slit in the plasmid DNA. So the endonuclease is otherwise called biomolecular scissor. Then with the in the circular plasmid DNA, extra chromosomal DNA which will replicate by itself, and in this the foreign DNA is inserted with the help of the enzyme called ligase. This combination of foreign DNA and plasmid DNA is otherwise called recombinant DNA. Now it is inserted into the Escherchia Coli, which act as a host, which contains DNA polymerase enzyme. Because of when the host is when the plasmid DNA with foreign DNA is inserted into the Escherichia coli host, it will by the DNA polymerase, it multiplies and many times, and we are getting insulin from that. The insulin is filtered out by the process of cleaning and filtration. We are getting the insulin. So by this way the insulin is prepared, and this the recombinant technology was accomplished by Stanley, Cohen and Herbert, Boyen. With this we conclude this video.